Okay, I'm aware that it's just me and one other talk separating you from alcohol and the networking bash. I will try to be short and punchy here. I'm Hilary Eaton. I'm the Chief Business Officer of Profluent. We are an AI company, and we're here to tell you about our mission to drive the drug discovery paradigm from one of random discovery to one that's more of bespoke design. So I don't think I probably need to convince this crowd that proteins are essential for many industries, including human therapeutics, which is this conference is focused on, but also agriculture, diagnostics, cosmetics, a range of things. And one of the intriguing things to me as a, I guess, former scientist turned to the dark side of business development is how many of our blockbuster drugs that exist today weren't actually designed for purpose. They were just random serendipitous discoveries. So for example, if you consider Cas9, which is kind of the workhorse in the industry for gene editing, that was actually initially discovered in a Dannon yogurt factory when they were taking a hard look at some of their fermentation and manufacturing processes. And how ironic that something that has the potential to fix broken DNA was discovered in a yogurt factory, right? So we'd like to do something about changing that. And so the goal of Profluent, we were formed from a premier AI research lab in Salesforce and OpenAI. And so our goal is to think about how do we translate ChatGPT's success from English language to protein language. Now, some models out there are really, at their heart, readers of biology. So if you look at AlphaFold 3, you're feeding a sequence in, and it's going to predict how it folds and therefore how it functions. What we're building at Profluent are models that instead write biology from scratch, and not some sort of like in silico version of random mutagenesis, but actually models that are exposed to billions of versions of naturally evolved proteins. And by exposing these models to that diversity, they effectively learn the grammar and syntax of protein language such that you can now treat these models pretty much like ChatGPT. Instead of prompting them to write my homework, an essay on Shakespeare's Macbeth, that has to be 2 pages long, you instead say, I have this therapeutic use case. To solve it, I need a protein that looks like this, that is this size, that does these things. Now write it for me from scratch. And it does exactly that. And I know that sounds like science fiction, but we kind of are in the era of science fiction. So what were the ingredients of ChatGPT, right? If you look back 5 years, your phone could not finish your sentence. And now we have apps that people are using to not only finish their sentence, but actually inform strategic decisions in their home lives and their work lives. And so really the 2 key ingredients for that success were access to large quantities of high quality data, and then the size and the architecture of the models, things like the transformer algorithm. Right? So then turning to the world of proteins, we're kind of blessed to have billions of examples that nature has just evolved out there. And so we've put together what we believe is the world's largest curated protein resource. This slide is actually slightly outdated. We were at 80 billion unique protein sequences last year, and we're now closer to 200 billion unique protein sequences. So when you look at the scale of that data and the curation of that data, that's more than ChatGPT 3, AlphaFold 3, et cetera, we're exposed to. It's just an insane scale of data. So now how about the models themselves? So in this case, bigger is better. Bigger models basically allow you to generate more valid sequences, they allow you to cover more diversity, and they allow you to capture more function. So for those in the audience who may not be AI experts, myself included, what does that kind of mean in layman's language? So if we take the same model architecture, but small, medium, extra large, all trained on the same data, and then set these models to all perform the same task. For example, maybe we say, you want a better protein that's more thermostable. Great. The small model works away, it comes back with its million sequences that it spit out, and you forensically look at what it did, and it took a domain from a heat shock protein, and it tried putting it in every single place in your protein till it got to a place where it didn't disrupt the function of your protein. And you say, good job, model, that's exactly what I wanted you to do, right? When you look at what the extra large model does, it'll spit out proteins where you put it into AlphaFold 3, and AlphaFold 3 says, doesn't fold, doesn't function, like this is craziness. And then you make it in the lab, and it folds, and it functions, and it's more thermostable, and we don't know why, right? So the models be having these emergent behaviors where it's actually like seeing patterns that the naked eye can't see, and then sort of working its magic. So, moving on, you can have the biggest, baddest model. That's super cool. What are you actually doing with it? And so we've tried to validate ours across the last year and a half across a wide variety of proteins, ranging from gene editors like nucleases, base editors, reverse transcriptases, programmable DNA binders, but then also moving into new areas, multi-substrate enzymes, biocatalysis enzymes, antibodies, et cetera, et cetera. So I'm going to take a minute and zoom into gene editing as an example use case that's particularly relevant to the cell and gene therapy audience. So, about a year and a half ago, when we were thinking about launching as a company, the board said, what would be a great use case for seeing if our model can write from scratch a pretty complex enzyme? And somebody said, how about Cas9? It's the workhorse of the industry, it's a pretty complex protein, and it has a lot of obstacles in the way of effective commercialization. And so we set the model with some pretty strong constraints where we kind of said, don't just go wild and go diverse as you can, but build something that you could compare apples to Cas9 that has the same PAM interacting domain, takes the same guide, et cetera, et cetera. And from that campaign came many molecules, but one we labeled OpenCRISPR-1. Now, because we have millions of assets, inherently any one sequence doesn't have a ton of value to us. And so we thought, we validated it in hex cells, but everybody knows hex cell plasmid delivery is far from the end of the road. So how about we just open source this molecule and see what people do with it as a research tool in their own cell types of choice. And so we took a page out of the tech playbook and said, what if we actually open source it? For free for commercial uses. And everybody's like, that's crazy, we can't do that. The liability is not software, but we found a way to do it. And honestly, we were shocked by the interest that the community had in it. In some of my former roles, I would be absolutely thrilled to have maybe 2 bidders for an asset. That would be fantastic, drive the price up. We've gotten over 1,200 license requests for this molecule. And we've talked to some of them and said, are you crazy? This molecule is not, clinically de-risked like Cas9, which is almost off patent. What would make you go for this? And the answer is sort of that it removes barriers, right? That it's something novel that acts like Cas9, but doesn't look like Cas9. And so it's had wide adoption across the industry. And of course, pertinent again to this audience is that now all those people who adopted the molecule a year and a half ago are coming back to us and saying, hey, who makes this GMP? And we're saying, uh-oh, that's not what we do. We're an AI design company. So we're forming partnerships as we speak to make sure that this is available to our customers as they take it forward. Now, our business model is clearly not just sell every enzyme for free, right? And it's not even about one molecule at all. It's about literally having every molecule at your fingertips. So in this phylogenetic tree shown on the right of the slide, what we have is in black natural Cas variants and overlaid on top of it green or AI-generated variants on top of it. And essentially what you end up coming up with is this sort of catalog to pick from where no matter what combination of criteria you're looking for in your editor, we have it. And we didn't have to sort of look accidentally through multiple metagenomes to try to see if accidentally nature had evolved the thing that you're looking for. We can just create it out of the gate. So some examples of things that have come out of this process include, let's see, we had a bunch of deaminases that we licensed to Revity last week. One of those randomly turns out to have a 1 to 2 base pair editing window. We don't know why. We're working on figuring out the mechanism, but it's awesome because then you don't get bystanders, right? And so when you have the ability to explore with AI sequence space that nature never got the chance to explore, you start getting novel, new to nature functions that sort of weren't out there before. So, it's not just sort of, we're not an editing company, we're not an antibody company, we are an AI protein design company, and we continue to innovate on the model side. So, in tech, everything moves fast. Our investors, are really used to sort of the move fast, break things, and that's not really biotech. Things are a little bit slower in human therapeutic development, but we've maintained this steady pace of as our models size up and as our database of training data sizes up, and as we figure out how to better pre-train, post-train, and fine-tune and align models, we moved from building simple proteins to building more complex scaffolds like OpenCRISPR and deaminases. We then looked at sort of precise functional control. So Pam got brought up in the earlier talk from LifeEdit, right? This idea that we do have a storied history of, okay, SpyCas9 evolved to land here, but I would like it to land elsewhere. So I will take my favorite hi-fi or whatever, I'll drop that Ph.D. into Cas9, oh no, it doesn't work anymore, right? And so what we took a look at is, can you look at every single Cas scaffold, natural or AI generated, every single possible PAM, and then figure out with no wet lab work which ones are compatible, and we did that zero shot. So that was published earlier on this year. Now that we've shown that we can predict using AI a relatively simple protein DNA RNA interaction, now we're moving on to more complex ones like programmable large-scale insertion, which really would be kind of the holy grail for both monogenic disease and for cell and gene therapy. So now transitioning a little bit to patient impact. Why do we care so much about democratizing gene editing with AI, right? When you look at sort of the panel of first-gen, second-gen, third-gen editing companies that are out there, the sad fact is that many of them no longer have rare disease in their pipeline. Or they did for a while, and then it got cut as soon as they had a clinical asset or something with a larger disease population to move forward with. And I mean, that's due to a lot of things in our current system that are fundamentally broken. It's not the fault of any of these companies, it's that we have obstacles in the regulatory path. Problems with trial timelines not being fast enough for kids that deteriorate too quickly, issues with high COGS to manufacture, and all of this leading to like economic unsustainability because either you're passing on an academic royalty that's super high, or the combination of all the things that are required for your complex product just leads to an unsustainable economic stack. And so, AI is not the magic wand that is going to fix all of these things, but it can help in certain domains. One is trial timeline, right? So when you look at this idea of, is there going to be a future where there's going to be personalized genomic intervention, and it's going to be not much different from a care team deciding on surgical intervention or whatever medicine you're going to take, right? One of the things that's going to matter is speed and cost, right? When a child is identified with a de novo mutation, how quickly can you develop a bespoke editor that is tailored exactly to that kid? And can you do that cheaply? And we believe that Profluent's platform can do both of those things. Maybe on the sort of like less sexy side scientifically, but from the BD aspect, compelling is that we are wholly owned in everything that we do, so we have no royalty to pass along. We just need to make a small sliver of protein, so we can do things at a really sustainable price. So again, don't want to leave you with the idea that the AI hype, that's the thing that's going to solve all of the industry's problems. It's not. It is a tool, right? But it is a pretty amazing tool when you combine it with minds that are experts across all of these different verticals, from machine learning to gene editing to protein structure and design and beyond. And we're lucky enough to have a panel of advisors that have overseen not only some of the most fundamental theoretical discoveries that have happened in natural language processing and machine learning, but have also translated it into commercial success within the tech world. So really lucky to have all of these folks in a room, and we've created sort of like that closed feedback loop where sort of our dry lab sends to our wet lab, sends back, and so we really try to not have sort of bottlenecks in moving that process forward quickly. And then finally, I'll end on a little bit of a call to action, right? So, we talk about rare disease pretty, like rare disease is that something that one person has, right? But when you aggregate all the folks who have rare disease in the world, it's 300 to 400 million patients. So I guarantee that everybody in this room either knows somebody or knows somebody that knows somebody who has a quote-unquote rare disease. For me, that's myself and my 2 young daughters, and that is sort of like the passion that I bring to this, right? We're here not just to make money for our investors, we're also here to bring cures to patients, whether they are, a snowflake of one or whether they're part of a larger patient population. And to do that, we're gonna have to break a lot of things about the system, but we're really excited to do that. So I will end early and give you some time back. Thanks.
Thank you very much, Profluent.